Hello dear students. Previously we studied that ATP and nucleotide triphosphates store energy that is released when the phosphoryl bonds within the molecule are cleaved. Today we will talk about other phosphorylated compounds which are found in the biological system, and their breakage also releases energy upon hydrolysis. These include biological phosphorylated compounds and thioesters. So let's take a look at the biological phosphorylated compounds, the first one of which is phosphoenol pyruvate or PEP. Maybe you remember something from glycolysis, during which phosphoenol pyruvate is formed. So phosphoenol pyruvate contains a phosphate ester bond that undergoes hydrolysis and yields enol form of pyruvate. This enol form of pyruvate can immediately tautomerize to a more stable form of pyruvate, which is keto form, meaning the reason is because the phosphoenol pyruvate has only one form, यानी जो reactant है phosphoenol pyruvate ये सिर्फ एक ही form में यानी enol की form में exist कर सकता है, whereas pyruvate which is the product it has two possible forms. ये enol की form में भी exist कर सकता है और keto form में भी exist कर सकता है. Therefore the product is more stabilized, यानी जो product है वो ज़्यादा stable condition में है as compared to the reactants. And this is the reason that the breakage of the phosphorus bond in phosphoenol pyruvate releases high amount of energy. So here you can see phosphoenol pyruvate, and by the addition of water molecule to phosphoenol pyruvate there is a release of inorganic phosphorus, and the molecule is converted into the enol form of pyruvate, which is spontaneously tautomerized to the keto form in biological situation. The process is catalyzed by pyruvate kinase enzyme, and the amount of energy released during the reaction is minus 61.9 kilojoule per mole. So now let's take a look at 1,3-bis phosphoglycerate, which is a 3-carbon compound, and it contains an N-hydride bond between the carbon 1 carboxylic group and phosphoric acid. So when in this anhydride bond of 1,3-phosphoglycerate there is addition of water, the compound undergoes hydrolysis, and with the release of inorganic phosphorus 1,3-phosphoglycerate is converted into 3-phosphoglyceric acid. We can see it here in this diagram that 3-phosphoglyceric acid is formed with the release of inorganic phosphorus, and this acid readily undergoes ionization, and here we can see a carboxylate ion which has got the resonance structures and is getting stabilized by these resonance structures, and here is the formation of 3-phosphoglycerate. The energy released in this reaction is also very high. We can see that this is equal to minus 49.3 kilojoule per moles. So most of you would be familiar with phosphocreatine. This is also known as creatine phosphate and can be denoted by PCR. Phosphocreatine serves as the mobilizable reserve of high energy phosphates in skeletal muscles and brain, and it helps to recycle adenosine triphosphate, meaning ATP. After creatine phosphate undergoes a certain number of dephosphorylation and rephosphorylation cycles, it breaks down and forms creatinine. This creatinine is then excreted in urine. At the moment we are interested to know that which bond is broken and it releases inorganic phosphorus. So here you can see the structure of phosphocreatine, and phosphoryl group is attached to nitrogen. So the breakage of bond between phosphoryl group and nitrogen is the one which releases creatine and inorganic phosphate, thus releasing large amount of free energy that can be used in the biological processes. So hydrolysis reaction has resulted here in the formation of creatine and inorganic phosphorus, both of which are stabilized by the formation of their resonance structures, and the energy produced is minus 43 kilojoule per mole. Now coming to thioesters. So thioesters are the esters in which sulfur atom replaces the position of oxygen in the normal ester, as you can see here. So the thioesters have also got large negative standard free energy change of hydrolysis. nature usually chooses to make esterification reactions using the thioesters. And the reason is that thioesters exist in a form which can be easily broken, and the energy to break the double bond is less as compared to the energy which is required to break the bonds in regular esters or in oxygen esters. This is because of the fact that the carbonyl carbon found in thioesters has got lower activation energy and it is more reactive than the carbonyl which are found in normal esters. The most important example of thioesters is acetyl coenzyme A, which is playing important role in metabolic functions. It has got the acyl group which can undergo trans-acylation reaction, meaning transfer of the acyl group from one molecule to the other molecule, or the condensation reactions or oxidation reduction reactions. The hydrolysis of acetyl CoA leads to the generation of carboxylic acid. So in this picture you can see that the hydrolysis of acetyl CoA forms acetic acid, and this acetic acid can be ionized and further stabilized by the resonance structure, and the energy released in the reaction is minus 31.4 kilojoule per mole. So this picture depicts that the amount of energy released by the hydrolysis of thioesters is far more as compared to the energy which is released by the hydrolysis of oxygen esters. This is because of the fact that carbon and oxygen atoms have got their resonance structures which are not found in case of sulfur. Now let's revise what we have learned. So for the hydrolysis reactions which release large negative standard free energy, we saw that the products appear in more stable condition as compared to the reactants, and this can be because of one of the following reasons. So the first reason is that there is some electrostatic repulsion forces within the reactants, because of which when the products are formed they appear in a better electrostatic condition. In other situations, the products are stabilized by the ionization within the molecule, or by isomerization or tautomerization, or by the formation of the resonance structures, and we have just studied these. And other important point is that the phosphate compounds which are found in the living organisms, they are arbitrarily divided into two groups, and this grouping is done on the basis of the amount of free energy which is produced as a result of hydrolysis. So first group is the high energy compound and the second is the low energy compound. Among the high energy compound are the reactants, or are the compounds for which hydrolysis releases energy that is more than minus 25 kilojoule per mole, meaning any compound which has got the free energy below minus 25 kilojoule per mole will be categorized under the heading of low energy compounds. And like we studied that ATP has got the free energy change of minus 30 kilojoule per mole, so it is a high energy compound, whereas the hydrolysis of glucose 6-phosphate releases minus 13.8 kilojoule per mole energy. So it comes under the category of low energy compounds. And here you can see in this table the standard free energies of hydrolysis of some of the biological phosphorylated compounds. With this lecture comes to an end. Thank you for listening.